Hi, my name is Saura, and I work on outreach and diversity efforts at the Halıcıoğlu Data Science Institute at UC San Diego.
Hello, everybody. My name is Bradley Voytek, and I am a professor of cognitive science and data science at the University of California at San Diego. I'm part of the neurosciences graduate program. I did my undergraduate research in psychology and did my PhD studies at UC Berkeley in neuroscience. And I've gone on in my career working in industry as a data scientist before coming back and working now as a professor, running a neuroscience research lab full of wonderful people. And this research lab has actually a mix of people, as you can see here, from cognitive science, which is broadly the field that is trying to understand what is intelligence, artificial and biological, as well as neuroscience, data science, and a variety of other different fields too. I have students in my lab from mathematics and so on. And so in my lab, we use a variety of different approaches to try and understand how is it that the 86 billion or so neurons in the human brain communicate with one another. And so this is my group here at UC San Diego, and we love brains. We are very interested in how this big, fatty, massive tissue in our heads gives rise to all of our complex behaviors and thoughts and all of the various interesting things that we are able to do. I find brains fascinating, and there is an incredible amount of opportunity here to use computational mathematical data science tools to try and figure out how all of these different aspects of who we are biologically come together to make us these incredibly thinking beings that we are. So how is it, this is what we are interested in my group, how do we figure out how brains work? And today I'm going to talk a little bit about some of the approaches that both my lab takes as well as many other labs around the world. So one of the first ways that we use to try and understand how the brain works is something called brain imaging. And brain imaging is a broad term for just trying to figure out how is it that we can look at the brain. What are the different ways of looking at the brain and its activity? And so as I said at the beginning, the way that brains communicate is we have all of these little neurons, brain cells in our head, 86 billion or so of them, and they communicate with one another using these little chemicals called neurotransmitters or neuromodulators. And those neurotransmitters and neuromodulators then cause little electrical signals called action potentials or spikes. And so the brain communicates using a currency of electricity. Now, the way that we study human brains is often by looking at it, what we say, non-invasively. So how is it that we can look at brain activity without having to go inside of the brain? And so one of these major methods is called fMRI, which stands for Functional Magnetic Resonance Imaging. And this is an indirect measure of brain activity. It's not actually measuring electrical signals. It's measuring a secondary byproduct. It's measuring changes in blood oxygenation and blood flow in the brain. So when groups of neurons in the brain are working really hard, they send a lot of these electrical signals, and that uses energy in the form of sugars, glucose, oxygen, and so on. And so in order to then replenish their energy stores, the brain gets an influx into that brain region of oxygenated blood. And this is what fMRI measures. And so this is an example of the kind of fMRI image that you might see a research group publish and talk about. So, for example, in the upper left here, this is what's called a coronal slice. It's looking at the brain from forward to backward. And the next image to the right of it is looking at it from sort of left to right. And then that bottom image is looking at it from top to bottom. And so we're looking at the three different dimensions of the brain here. And you can see there are certain areas that are lit up, meaning there's more activity, more change in oxygenation in those brain regions. And this might be a group of people that are sitting in a brain scanner, maybe doing like a memory task. They have to remember a list of numbers or something like that. And when they're remembering this list of numbers, you see an increase in this activity in these front parts of the brain, cleverly called the frontal lobes. Another way that's older that we have for brain imaging is called EEG, which stands for electroencephalography. So electro meaning electricity, encephalo meaning head, brain, graphy meaning the measurement of. And so this dates back almost 100 years. Into 1929 is the first EEG, non-invasive, used to study human brain activity. And so in EEG, here's a photo of me with an EEG cap on. In EEG, you stick these electrodes on the surface of the head, and they record the electrical activity coming off of these millions and millions, billions of neurons in your head. And so these traces here, the top trace next to this guy, Hans Berger, who is the one that invented the EEG system in 1927 or so, that top trace is electrical activity from the back of the brain. In the occipital lobes, which are the visual parts of the brain. And the bottom trace is just a 10 hertz, so 10 cycles per second sine wave. And you can see that the actual brain activity has this oscillation at around 10 hertz. And so this was one of the first things ever discovered about human brain activity. Now, what's interesting is this non-invasive measurement of human electrical brain activity is very indirect. It's very noisy. And so, like I said, you've got all these electrodes in the top of your head, but there's 86 billion neurons underneath those electrodes. Well, hopefully I have that many in my brain. And all of those electrical signals coming off of all of those neurons sort of hit the inside of the skull, and like a wave crashing into a rock wall, just kind of splash and smear out. So EEG is fantastic at figuring out when something happened in the brain, down to a millisecond resolution. You can see it changes in brain activity, but you can't tell where that signal is coming from, and that's because, like these waves crashing into the rock wall, all of these electrical fields from all of these neurons hit the inside of the skull and sort of smooth out and smear out. fMRI, in contrast, is fantastic, as you saw from those first brain imaging pictures that I showed you, at seeing exactly where something in the brain changed, something happened, but not when, because these changes in blood oxygenation that fMRI measures are very slow. They happen like 5 to 10 seconds after the brain was active. And neurons send an action potential and are finished within a millisecond, so 1,000th of a second. So fMRI is great at saying where something happened in the brain, but not when. EEG is great at saying when, but not where. So they're complementary in that sense. So both of these have those kinds of drawbacks, if you can know where but not when, or when but not where. Now, one way that my lab gets around these drawbacks is we work with people who have undergone certain kinds of brain surgery where a big piece of their skull has been removed. And the reason that we work with these people is because they now have a literal window into the brain. The skull is no longer in the way. It's not blocking our signal. So in these people, these recording electrodes that we have in scalp EEG, they're actually implanted directly into the brain as part of the standard of their clinical care. So a lot of these patients with whom we work have epilepsy, which is a disease that causes seizures. For the vast majority of people who have epilepsy, they can take anti-epileptic drugs which suppress their seizures. A seizure is, in the most severe form, causes the whole body to become very rigid, and people lose consciousness after, they might have some violent shaking even. And in the most severe cases, this can happen multiple times per day. As you might imagine, this is not very good for the person's quality of life. Now, thankfully, for the vast majority of people that do have this disease, anti-epileptic drugs are sufficient to keep their seizures under control. But for whatever reason, a small percentage of people that have this disorder, anti-epileptic drugs don't work. And so they will choose to undergo a surgical procedure wherein a piece of their skull is removed, and then that piece of skull stays off for sometimes 2 weeks while the patient is hanging out in this epilepsy monitoring unit here at UC San Diego. The neurosurgeon with whom we work is Dr. Sharona Benheim. And what Dr. Benheim does is, while these patients are sitting in this hospital epilepsy monitoring unit, she's implanted these electrodes directly into their brains to identify the exact part of the brain that is pathological. That is, what is the part of the brain that is causing the seizures? And in order to identify that, none of the non-invasive brain imaging tools like fMRI or EEG are sufficient. And so Dr. Benheim and all these other neurosurgeons around the world, they have to invasively implant these electrodes and wait for a week or two for the person to have enough seizures that the surgical team is confident they know where the seizures are originating from, and they can surgically remove the part of the brain that is causing the seizure, known as the epileptogenesis zone, or epileptogenic zone, which is just epilepsy genesis, or the start of epilepsy. And then they put the skull back in, and the patient is almost always, if not cured. They have significantly fewer seizures and less severe seizures. Now, this particular image from one of my past research projects is actually not from a group of people who had this kind of brain surgery for epilepsy. These are people who had traumatic brain injuries. that causes a swelling of the brain. If you get an injury like a big bruise or something like that, it might swell, maybe even for a couple of days if it's a very severe injury. That's normally not too big of a deal. But if the brain swells, the brain doesn't have anywhere to swell to but into the skull. It sort of swells and hits the skull. And unfortunately, all of the major arteries carrying oxygenated blood from the heart to the brain sit on the outside of the skull like this. And so if the outside of the brain like this. So if the brain swells into the skull, it pushes those arteries closed and cuts off blood supply to the brain, which is not great. So this was done in collaboration with a surgeon at UC San Francisco, Dr. Manley, and he did these surgeries where they remove a big piece of the skull, and that gives the brain a place to swell to that is much safer. And so these people have their pieces of skull off for several months. And this surgery is known as a decompressive hemicraniectomy. Decompressive meaning trying to reduce the compression, and hemicraniectomy, hemi meaning half, crani meaning sort of skull, and ectomy meaning removal. So it's a removal of half of the skull in order to reduce compression. Now, in this particular case, these patients, the skull contains living tissue in our heads, and these patients want their skull back at the end of the swelling period of several months. And so this is an image from a news story several years ago of a woman who had one of these surgeries, and she shared her story about this. And you can see here on the left, this is what it looks like during the several months when the skull is off. You can see that right half of her head left on your side of the screen here, sort of sunk it in because the skull is no longer there. It's just skin, and then the brain underneath. And in order to keep the skull alive, the tissues alive, what the surgeons will sometimes do in this middle image you'll see here is they actually implant the skull into the abdomen to keep it essentially bathed in the body's own fluids. It keeps it alive. And then after the several months of the decompressive period, once the swelling has gone down, they remove that piece of the skull from the abdomen and reimplant it into the head, sew everything back up, and it's phenomenal. I mean, neurosurgery is amazing. This is somebody that prior to the advent of this surgery in common practice 15, 20 years ago wouldn't have survived her initial head trauma. And so now what I'm going to show you, this is warning here, is because I'm going to show you a video I've taken from the operating room of somebody undergoing one of these brain surgeries. And I give you the warning because I teach very large classes here at UC San Diego to several hundred students, and I've had students in the past faint because this is an image of an open brain surgery. Nothing too gory, but just, if you're the squeamish type, I'm going to give you a countdown to tell you to look away. And so here it is. Look away in 3, 2, 1. Now I'm playing the video here. Okay, for those of you who didn't watch, you can look back now. And so this is a video I took in the operating room of one of these patients undergoing one of these surgeries. And you can see a couple of interesting things, I think, that may be surprising. First of all, the brain sort of pulses a little bit. Even though the brain is only about 2 to 3% of the overall body mass, it gets about 20% of the output from the heart. Brains are we say, metabolically expensive. They require a lot of energy in order for them to operate compared to their size. And so because of that, they get a lot of blood with every heartbeat, oxygen from the heart and lungs. And so because of that, they sort of pulse with every heartbeat. Another thing you might have noticed is that there are little pieces of like plastic on the brain with these numbers written on them. So the patient is actually awake during this part of the surgery. So everybody's brain is a little bit different. Roughly, if I point to the left front part of your brain right about here and ask somebody who knows a little bit of neuroscience what that does, they'll say, oh, that's maybe Broca's area. It's involved in speech production. If I point to the back of the brain, I could say, oh, that's vision, and we'd say auditory and motor control and sensory perception and so on. So we roughly have, all the different parts of the brain mapped out and related to different functions. But everybody's brain is a little bit different. And so if it so happens that a patient has epilepsy where the part of the brain causing the seizure is anywhere near a part of the brain that is responsible for controlling speech and language or controlling our body movements, the surgical team will usually opt to wake the patient up during the surgery And in this case, the patient was hearing a list of words, nouns specifically. And so you hear chair, and their job is to generate a verb related to that noun. So if I say chair, you might say sit. If I say train, you might say ride. And while the patient is doing this, while they're awake laying there with part of their brain exposed, one of the surgical team has a little electrical stimulating device, and they're stimulating different parts of the brain. And so if the patient is sitting there and they're doing this task, chair, sit, train, ride, and one of the surgical team electrically stimulates one of the brain regions, and the patient instead says chair, sit, train, ride, what they'll do is they'll mark that part of the brain physically with a piece of plastic temporarily that says, a number on it, like 47. And they'll write down in their notes that stimulation site number 47 caused speech arrest. And so they know that when they're trying to go through the parts of the brain to identify and surgically remove the pathological part of the brain causing the epilepsy, they don't go through one of these areas that causes something like speech arrest, because they don't want to damage that brain tissue, those neurons, because they don't want it so that when the patient wakes up, they're unable to speak. Because the recovery time from that can take months, if not years. And so all of this is to say that there's a lot of very interesting things that we can learn from how the human brain gives rise to cognition and intelligence by working with these patients. So this is a video from a friend and collaborator at Stanford University, Yosef Parvizi. Dr. Parvizi is an epileptologist, and he works with some of these patients. And in this particular example, this is from a publication he had in 2012. He presented this video of one of the patients who had electrodes implanted for this kind of epilepsy surgery in a part of the brain known as the fusiform face area, which is a part of the brain that we know is related to our ability to perceive faces. And Dr. Parvizi in this experiment shows what happens if you electrically stimulate and disrupt this fusiform face area in a person. So I'm going to jump ahead here. So this is the patient, and he's going to be given a sham stimulation. So a sham stimulation means that it gives the click as though the electrical stimulation was provided, but it's fake. It's not real. And so it's a scientific control that is used to make sure that for all the patient knows that their brain received this electrical stimulation, and he heard the click, but no actual electrical current was applied to the brain, and he says, "Oh, I didn't. Nothing happened." And now here is this next part. Real current. And so if you, in this person who has these electrodes implanted in their brain, in this fusiform face area, electrically stimulate that part of the brain, which we suspect is related to our ability to perceive faces. If you disrupt it briefly through electrical stimulation, you have this interesting phenomenon where the patient says, "Well, it looked like you morphed into somebody else for a second. That was a trip." And so this is one of the ways that we use to figure out how the brain works. Now, as I said at the beginning, my lab instead, we take a different approach. We use math and data to figure out how brains work. And so we do these very carefully controlled kinds of experiments, like I showed you from Dr. Parvizi. But we take a sort of another perspective as well. What can we do with large amounts of data related to human brain activity? This is a paper, a project from another friend of mine, Brian Paisley, where you can see here on that brain image, there's these electrodes implanted over this particular part of the brain, the left-hand side of the brain, left-hand side. This is the auditory parts of the brain. And so what they did in this study is they gave people sounds. They presented sounds to them, acoustic waveforms, words, very specific words. And then they looked at the pattern of brain activity. And so here on the bottom left, this is a representation of sound where time is on the x-axis here, and frequency is on the y-axis. So very low frequency parts of the sound, ooh, versus high frequency, I can't go very high, and everything in between. And the color represents how much energy there is at the different frequencies. And so this is one word, one snippet of a word here, 2 seconds, it's spectrogram. And here they present that word, and on the right-hand bottom here, they have the brain activity. And so what they're building here, what they're trying to do is, given an input, which is this sound, and the output, which is the brain activity, can they construct a model that maps the input sound to the brain activity? And so one way of doing this is what's called through linear regression. This is a very simple model, which is a lot of people are somewhat familiar with it. It's basically fitting a line. So the equation for a line is y equals mx plus b. You could do this on huge amounts of data and use this to try and build a mathematical model that says, given an input, I want to know what the output is. And once you have that model built, you can invert it. So then you could say, if I present a new sound to somebody that I haven't previously shown them and record their brain activity, can I then pass it through this mathematical model, invert it, and try and guess what the sound was and play it back? And so what you're going to hear here are snippets of the words that the person heard, and then the reconstructed model based upon the patterns of brain activity of what Dr. Paisley and his collaborators thought the people were hearing. So there's the real world, and that's the reconstruction. Real world, reconstructions. So what that means is that you can kind of read brain activity. You can take some pattern of brain activity, and then you can guess what a person was hearing. And this has been done across different domains by many other research groups over the world. So given a pattern of brain activity, can you predict what they were seeing, for example? And this isn't just a really cool thing to be able to do. This is incredibly useful in terms of improving patients' qualities of life. So for example, if you have somebody who is paralyzed, that means they can't move because maybe their spinal cord has been severed. If you record the activity of their brain from the motor parts of the brain, so these are the parts, motor meaning the sort of body control, the movement parts of the brain. You record the activity from the parts of the brain that are sending signals down to the muscles in the body to tell them to move. Those signals, if the spinal cord has been cut, don't get through. So the brain is still trying to tell the body to move, but because the actual wire in the spinal cord that carries that information from the brain to the muscles has been severed, that information can't get out. Can you take that pattern of brain activity, decode it using some of these kinds of machine learning tools like linear regression and more sophisticated versions of regression, and then instead of the person moving their body, send that decoded signal to robotic effectors to have the patient move robotic body parts instead. So these are the kinds of things that neuroscientists around the world are working on. And so in the last couple of minutes here, I'm just going to briefly tell you about my path as a data scientist. So at the very beginning, I said that I did my undergraduate work in psychology. I thought I was become a therapist. But then I fell in love with the brain and brain science. So I did my PhD in neuroscience. And then after I finished my PhD, I got recruited to work as the first data scientist at Uber, back when it was still called Uber Cab, a very small 10-person startup in San Francisco at the time. Because I had done a lot of mathematical and computer programming, data science related work during my PhD research, and I was asked to help Uber at the time build out their data science team and data science strategy. So how does a private company like Uber, which is a ride-sharing service where you open up your phone and you request a ride from a driver or want food delivered, how does a company use that huge amounts of data that they're collecting in order to make the system and service better? So if you want to order some food, you want the food to get delivered to you pretty quickly. If you order a ride, you want the ride to get there to you pretty quickly. And so one of the ways that companies do this is by using huge amounts of data. So, for example, if we want to make Uber more efficient, we don't just have to know what are the most popular times of day that people take rides. So you're much more likely to see people have a huge spike in demand. Friday and Saturday nights are very popular on the weekend. And the lulls are 2 to 4 a.m. on like a Tuesday, very low demand. So it's one thing to know those historical patterns as well as how those temporal patterns of demand change as a function of space. Certain parts of the country have more people, more population. Certain parts of the city are more popular destinations at different times of day and different hours of the day and days of the week. But also you have to know, what are the weather patterns going to be? If it's raining, people are more likely to take a ride-sharing service. You don't just have to know that, you have to know when are concerts happening. And also how popular are those concerts likely to be. And so you can find scrape data about upcoming concerts, and then find the people performing those concerts and scrape data on the number of Instagram followers they have, which is a good proxy for the popularity of the concert. But not only that, but if there's like a sporting event going on, a basketball, football, baseball game, in particular, let's use the example of baseball. Baseball has 9 innings. And if by the end of the 5th or 6th inning, which is more than halfway through the game, the home team is losing by more than 4 or 5 points, people start to leave the game early. So you have to know not only what sporting events are happening when, but also how badly is maybe the home team being beat, because you know that there's going to be a spike in demand around that baseball stadium if the home team is losing by more than 5 points at the end of the 6th inning. And so all of this data and all this information gets aggregated together. And so here in the last few minutes, I'm going to talk a little bit briefly about what does the future of data science look like. So this is, again, a little bit old. It's about 10 years old. This is a film clip from the film Batman Begins of just Christian Bale, who plays Bruce Wayne in that film, lying down. And this video has been processed by this group of researchers in order to amplify very subtle movements in the film image. Now, this algorithm that they created for amplifying subtle movements was actually created in order to observe prematurely born babies in neonatal ICUs, NICUs, to monitor their breathing and heart rate. These little tiny premature babies, it's very important to monitor their breathing and heart rate 24 hours a day. Problem is, the ways that we have for monitoring that is pretty invasive. You got to like touch the baby and poke them and prod them, and it wakes them up and it disrupts their sleep, which is not great for their long-term recovery. And so they created this algorithm to film the babies and then amplify subtle movements in order to see if they could monitor the baby's breathing and heart rate from film. But then they took that algorithm and they applied it to existing movies. So you can see there Christian Bale's pulse from this film clip. Which, okay, pretty innocuous, no big deal. But now imagine if like TikTok or Instagram or something has these algorithms in place. What happens if you walk into a party and you see somebody that you like? Your heart rate starts to increase, right? Can you use these kinds of algorithms on existing data in order to identify maybe certain things about us and our behavior that we don't want other people to know? Or in the more extreme, they took this algorithm and they applied it also to, in this case, a bag of chips. So just like we can build a mathematical model to reconstruct the sound that a person is hearing based upon their pattern of brain activity, here they are reconstructing the sound that is changing the movement on this open bag of chips using similar mathematical models. So what this means is, if every time I speak, the way that speaking works is that I am pushing sound out of my throat, pushing air out of my throat, which then that air moves and hits somebody's eardrums inside their ear and vibrates their eardrums. But that sound isn't just vibrating eardrums for people to hear, it's vibrating everything around us. Leaves on a plant, a bag of chips. And so the math that we can use for something like trying to reconstruct what somebody is hearing or somebody's intent to move in order to help them clinically can also be used, the exact same kind of math, to turn a bag of chips into a visual microphone. A little hard to hear, but you can hear maybe Mary Had a Little Lamb being played. And then they amplified this bag of chips using their algorithm and then did this mathematical reconstruction. And so this kind of technology is pretty amazing, but the underlying math, the underlying ideas are very similar across these different fields. And so this leads me to ending on some of the stuff that my lab does, which is neural data science. So data science related to the brain. And so, in brief, one of the things that my lab is interested in is all of the different components that we know about the brain. So for any arbitrarily tiny little part of the brain here, this is a little boxes around a part of the brain known as the dorsal lateral prefrontal cortex. Dorsal meaning sort of like top, like a dorsal fin on a fish. Lateral meaning to the side, so dorsal lateral. Prefrontal meaning it's part of the frontal cortex. It's a little bit prefrontal is a little bit farther forward. So dorsolateral prefrontal cortex, cortex meaning sort of the outer part of the brain. Cortex literally means bark, where all of the neurons tend to exist. And so for any little patch of the brain, we know a lot about here on this list on the left, it's architecture, meaning how thick is the cortex there? Some parts of the cortex are thinner, some are thicker because they have different kinds of neuronal densities. What does the white matter connectivity look like? So white matter is the fibers that connect neurons to one another. And so what are the inputs and outputs to that part of the brain? So there's dozens of labs around the world focused on each one of these things using brain imaging methods, invasive and non-invasive, to study these things in the human brain. But we also know the different cell types, different kinds of neurons. There's 100 and something different kinds of neurons, depending on how you want to count them. And they all have a different morphology, meaning shape. They have arborizations, it's called, branches and dendrites coming out of them. There's different genes, 20,000 or so different genes expressed in different parts of the brain differently. They have different receptors for different neurotransmitters and so on. And so my lab is interested in taking all of these different kinds of data and bringing them together. So just like we might be able to take the number of Instagram followers that Beyonce has and the rain that's occurring or not, the weather in San Francisco, and any sports happening in order to try and predict better demand for Uber, we can take these different kinds of data about the human brain and all of these different maps that we have, and we can bring them together to try and do new things. And so if we can integrate brain structure, function, electrical activity, and all of these things, we can bring them together into a unified space that allows us to do some nice, simple mathematical modeling of them to try and make predictions. That is, can we find gaps, missing connections where topics should be related, but we currently don't have any information about them. So if I take brain maps related to Alzheimer's disease and I say, show me all the different parts of the brain that we think are implicated in Alzheimer's disease, which is a very debilitating, pretty awful disease that happens in usually much older adults, and that causes pretty severe memory loss. And we say, okay, here's the parts of the brain that we suspect based on other studies that are involved in Alzheimer's disease. Now take these other maps that we've brought together into the same space and say, are there genes that are statistically over or under expressed in Alzheimer's specific parts of the brain about which we may know nothing? That is, by combining these different kinds of data sets together, we can mine through them to find these potential missing links. That is, we can use the data that other groups have been collecting for generations now to help us generate new hypotheses to speed up medical care. And so, with that, I'm going to stop speaking. Thank you all for your attention. Data science is amazing. And the critical component here is taking all of this mathematical knowledge of data science and machine learning and applying them to specific different domains that will help you make new breakthroughs. All right, goodbye.
Well, thanks, Brad, for a great presentation. My name is Saura Naderi. I work at the Halıcıoğlu Data Science Institute, and I will be sharing some example activities that use linear and polynomial regression. These are mathematical models to find patterns in data. So first, let me share with you that math is one of the three main foundations of data science alongside computer science and domain knowledge. For smaller data sets, for example, 10 to 20 data points, you can solve the correlation between the variables by hand. But for large data sets, you'll want to use a computer to find patterns in your data set, which is where computer science becomes valuable. Domain knowledge is another way of saying subject matter expert. For example, Brad just shared about brains, and he has a background in psychology and neuroscience. If your data set is about biological phenomena, then you should probably have a strong background in biology. Is it about the economy? Is it about pop culture? Having knowledge about what the data represents allows you to have an intuition about the patterns that may be found in the data set. Now, here is an example activity that we call data-driven robotic art. It's a very creative way to dive into data science. This uses linear regression, and it may give your students insight into how data science is used to find the correlation between two variables and use that correlation to make predictions outside the data set. For example, the correlation between housing size and cost, or height and weight. To do this, we use simple linear regression. Once we find the linear regression model for the data set given, we use that model to drive these servo motors. Now, in this prototype, you'll see that there's a correlation between pollen on strawberry flowers and the size of the strawberry. And the servos are actually hidden behind the bee and the strawberry bush. Here's another visual of linear regression equations with different coefficients. You can see how the servo movements are slightly different depending on those coefficients. All right, so what kind of materials do you need for this? Oh, here we go. You'll need a laptop. You'll need 2 regular servos, an Arduino software, 6 wires, a cardboard platform, a USB cable, a hot glue gun, cardstock, paint, and some crafts. You can find information about how to do this activity on the NSF-funded AI Institute TILO's website under the Outreach tab. Here you'll find this portion of the presentation and the code. Now, if you've never used Arduino before, there is some setup required, but it's been around for about a decade. So if you just Google search, like getting started with Arduino, you should find a resource that speaks to you. The next thing you'd like to do is actually find a small data set that has an obvious linear relationship. You might need to graph your data points to make sure it does. For example, something that looks like this, but not something that looks like this. Now, to find the coefficients for this equation of the line, you'll need to minimize the sum of squares of the data points. And to do this, we can use these equations to find those coefficients. And then once you find those coefficients, you'll take the Arduino code that will be provided and simply replace the coefficients in this line here. Now you upload that code to the Arduino and visualize how the servos move. And once your students are done with that, they can decorate the board to visually represent the data set that they were using. So here are some examples of students creating their own data-driven robotic art. People did things like the correlation between how many hours you studied and how well you do in a test. Another tool that's pretty cool and actually created specifically for this presentation, which I think is really rad, is this polynomial regression tool. Now, what it does is you'll have a graph and you'll be able to click on the graph and these different data points, and you'll see the line fits the data immediately. And the default is 1-degree polynomial. And just to see the equation, you can run the polynomial regression and it'll give you the equation. And if you add more data points and run it again, you'll see how the coefficients are going to change. Now, what happens if you increase the polynomial? This is what happens. This is so cool. And it curves now because we have a different order of polynomial. So you get to immediately see how changing the math will actually better fit the data. Now, if you increase it again, didn't quite click it at that point, you'll see that the line will eventually turn into like an S shape. Now you can keep increasing the polynomial and see what happens. It's a pretty cool tool. It's a really simple way to visualize how the math actually is, it visualizes it. See the line fitting the data. Look at that. And then if you go crazy with it, you could see that it's like a really crazy line. And this is actually called overfitting. It might not be the best thing to do. Now, to find this tool, you can go on another NSF funded website, encore.ucsd.edu under Outreach, and you'll find that the polynomial regression tool. Thank you. Hello.
Hello, everybody.
We are here to answer any questions that you may have.
Yeah, we only have about 5 minutes left, so please go ahead and ask us anything, and we'll try our best. Saura, can you tell me a little bit more about the tool that you all built?
The polynomial regression tool?
Yeah.
It was built by 2 grad students that are working for the Encore grant. Yeah. And they made it so that basically you could just visualize how the line fits the data, and you can change the polynomial. And then every time you click on that button, it shows you the equation, which I thought was really neat, because it was a kind of aha moment of like, oh, that's why you would do that, for me. So I thought it would be nice to share.
Yeah.
If anybody has any feedback, I mean, we could always try to make adjustments to it too.
Right. Yeah, but it's very fun to play with. It's very intuitive.
Yeah, I think they did a really good
that kind of
job.
math is so fundamental to a lot of machine learning. You learn the basics and you build from there.
Stephanie asked if there's more pollen if they're bigger strawberries, and according to the data that I found, that was the case.
Would you recommend math teachers connect with CS teachers to find ways to bring into the classroom? I don't know if you actually necessarily need to find a CS teacher, but when you can use the programs and code to bring the math to life, like Saura and her team did, I find that to be very, very helpful. And so some math teachers may know how to do that already. And some of the tools that Saura and her team has built allow you to do that without really having a CS background. But I feel like bringing those two different teams together, it's always good to brainstorm and diversify. And when you bring the different backgrounds together, you come up with really fun new ideas. So I'm always a fan of collaboration, so I would say yes, but you don't strictly have to, depending upon your educational goals. To build off of what Linda Adams had said in the chat about bringing the two different groups together, I would even take it a step further and bring in colleagues and friends who work in the social sciences and even in English. One of the great things about the data science approach to things is quantifying things that may otherwise not be intuitively quantifiable. So on the English side, for example, there's a lot of work, as people are a little bit more familiar with, in large language models like ChatGPT, and that's quantifying language. And people have actually quantified word use and verb evolution in English by looking at how every book that's ever been written almost in the English language over the last 1,000 years, how verbs change over time and things like that. And that's through these kind of data science approaches. So it could be fun even to bring in colleagues outside of math and CS to sort of brainstorm data science ideas. Stephanie asks that, do you think teachers who aren't in math science should see potential for using data or data science in their classes? Yes, exactly. If so, how do you think other content areas can use data and data science? So the classic example that I was talking about is looking at verb evolution. So English has a lot of irregular verbs, right? If verbs were regular, we'd say I am, you am, he, she, it ams, right? Like I swim, you swim, he, she, it swims. Except we say, instead of that, we say I am, you are, he, she, it is. And that's an irregular verb. It doesn't have a regular structure. And people have shown that by text analysis of books written in the English language over the last 1,000 years, you can see a mathematical relationship where the more frequently a word is used in spoken or written language, the more robust it is against regularization. So in English, we have regular and irregular plurals. We teach students that the way that they make a plural is by adding an S, but that's not always true. We say oxen, and fish is plural of fish, for example, and that comes from the Germanic roots of English. And it's the same kind of idea where the more frequently words are used at certain points in time, the more robust they are to regularization. And so there's a reason why our plurals are irregular for words like ox, child, fish, goose, deer, and so on, because those were things that were common in life when the Norman invasion happened of England, and the people who spoke Germanic languages found it very difficult to learn all these special cases. And so those are done through sort of data analysis of text. So anyway, we are almost out of time. I think we're about to kick out. So I'm sorry if I missed Felicia's question, but I'll do my best. Felicia, have them email me, or you can email me and I'll send you links.